And our next speaker is going to be Fadi Jacob. Fadi, you're welcome to come on and share your screen and unmute yourself. He's an MD PhD student from Johns Hopkins. Hi there. And he's going to tell us about SARS-CoV-2 neurotropism that he's observed in the choroid plexus epithelium. Interesting reflection of Madeline Lancaster's observations earlier today. So looking forward to that. Thank you.
Okay, it's great. Thank you for everyone attending and inviting me to give this talk. For those who don't know me, I am Fadi Jacob. I'm an MD PhD student at Hopkins. I was working in the labs of Hongjun Song and Guo Li Ming. And today I'm going to give you a bit about recent work we've done looking at human-induced pluripotent stem cell-derived neural cells and brain organoids to really look at the potential for SARS-CoV-2 to infect brain cells. So just to give a brief background, a lot is known. So although the SARS-CoV-2 virus primarily seems to affect the lung epithelium as well as the vasculature, infection has been reported in other tissues such as the kidney and the intestine. I think a lot of clinicians now are increasingly appreciating that a lot of the extrapulmonary symptoms might actually be due to systemic sources of infection. So here, neurological complications at least have been associated in at least 30%, if not greater, of patients who have COVID-19. And this includes encephalopathies, things like anosmia, like losing your sense of smell, as well as cerebrovascular disease. And a lot of people have looked at clinical data and looking at postmortem tissue of people infected with COVID-19. And people have seen that you can see viral RNA in the brain and CSF of some patients, but most of these studies actually can't detect these things. So it actually remains pretty unclear whether these neurological symptoms are due to direct neural infection or is it just some sort of systemic process. And I guess another caveat here is by the time you look at postmortem tissue, this is usually weeks later than the actual initial viral infection occurs. So just because you're not seeing virus, it does not necessarily mean virus was not there infecting cells and is now just gone. And what was left is sort of the result. So knowing this, I think, especially for the brain, it's really important that we come up with a model system where we can actually track from the beginning of infection and look at sort of all the consequences leading from the beginning. So we're not stuck with postmortem tissue. So to do this, our lab does a lot with generating different brain cell types from human-induced pluripotent stem cells. In particular, we focus a lot on generating brain region-specific organoids. So to look at the SARS-CoV-2 neurotropism, basically we took neurons, astrocytes, microglia, and monolayer and infected them, as well as brain region-specific organoids for both the cortex, which is mostly dorsal forebrain, the hippocampus, the hypothalamus, and the midbrain. And so looking first at neurons, we culture these iPS-derived neurons on top of astrocytes. And this is looking about 120 hours later. And we use about 10 to the 5 titer virus for this initial screen. And neurons, we noticed you can get very, very sparse infection in monolayers. So if you notice here, you can find one cell labeled here, which with the SARS-CoV nuclear protein, which colocalizes with MAP2. But overall, if you quantify this, the number of neurons infected is far lower than 0.1%. We see similar things for astrocytes. So also looking at the nuclear protein on top of GFAP, we see very few cells, which is less than 0.1%, and we're actually able to replicate this result using primary astrocytes, where we saw similarly low levels of infection. And then lastly, we also looked at the iPS-derived microglia, which are marked by this PU.1 protein, and interestingly, we didn't see any microglia infected. So based on the monolayer results, we see that neurons and astrocytes can be infected, but it seems to be quite low, and microglia can't. So moving on to the brain organoids. So for all the brain organoid studies, we always do 3 conditions. One is treat with the SARS-CoV-2 virus, wait 24 hours. We also wait 72 hours, and then we have a vehicle, which we wait 72 hours after. And as you can see, across all these different brain regions, you can get infection of cells. So quantifying this, for most of these, we used at least 2 lines. And you can see you do get more infection. So it's about 1% across these brain regions, give or take a bit, but it is still quite low. And so to really determine what cell types are being infected, we co-stain with DCX, which is a neuronal marker, and the majority of these cells are neurons. But that's not to say with these brain organoids very early on, around 50 days, where we usually do these experiments, you don't really get so many astrocytes. So the brain organoid is not so good at looking astrocyte infection, at least as far as what we look. And so the really interesting finding we seem to see is in our hippocampal organoids, a lot of times you'll have a corner or maybe a portion of the organoid will actually differentiate into Cory Plexus. and this is marked with the marker TTR. And what we notice is in these regions of TTR expression, we had much more infected cells. And then looking closer at this, we also looked at the double-stranded RNA, which when the virus replicates, they produce these double-stranded RNA particles, and these cells just seem to be packed with them. So this was very interesting to us, but for reasons because of the hippocampal organoid protocol doesn't consistently generate Cory Plexus, so it's very hard to do more with this. So fortunately, over the past couple of years in the lab, I've been working on refining protocols to generate different regions of the mediopallium. So the mediopallium is a region of the telencephalon along the medium wall, and this consists of regions like the hippocampus, the cortical hem, and the Cory Plexus. And a lot of work determining how these regions are specified has been done in the literature, and especially a Society's group in the past were actually able to take human embryonic stem cells and differentiate them to these different regions. And basically, what they found is by modulating different Wnt and BMP signals, you can specify these different regions. So using these findings in the lab, I've been able to refine and generate protocols for organoids where you can generate mostly cortical hem, hippocampus-type tissue, as well as the Cory Plexus. So specifically for the Cory Plexus, I've moved to using a feeder-free platform. So basically, you can grow the stem cells in feeder-free conditions. You aggregate them using an aggregable plate. And for this protocol, I like to use about 5,000 stem cells per organoid. This way, you get very reproducible embryoid bodies. And so these embryoid bodies we pattern to neural ectoderm using dual SMAD inhibition, as well as Wnt inhibition to really push more towards anterior fate. And from there, we use very high Wnt agonism using the GSKB antagonist CHR, as well as we use BMP signaling using BMP7. We treat over a long period of time. It's about 22 days. And as you can see during this treatment, these very round EBs start to form these extensions, which by 50 days in vitro form these very small, fluffy extensions of cuboidal epithelium. And using markers, we can determine that this is Cory Plexus. So OTX2 is a transcription factor which marks Cory Plexus. So this in combination with aquaporin-1 and the TTR is actually very, very specific for Cory Plexus cells in vitro. And if you look at the morphology, it actually resembles the epithelial projections here. And we use two lines here, and you get basically around 90% purity for these cells. So looking further, we also compared the transcriptome. So we took the Cory Plexus organoids, we did RNA sequencing, we compared them to the hippocampal organoid, as well as we took already published data for human Cory Plexus and just compared this. And what was great was that we found that the Cory Plexus organoids were very much enriched for these Cory Plexus markers, as well as adherence junction markers, signaling that's known to come from the Cory Plexus like BMP, as well as a whole range of these ion channels and solute transporters that are very important for CSF function and production. And this was all confirmed by qPCR as well. And so interestingly, we know the SARS-CoV-2 virus is able to infect cells because it binds to the ACE2 receptor. So we actually compared the expression of ACE2 between the hippocampal organoid and the Cory Plexus organoid. And as you can see here in the hippocampal organoid, you do get some expression of ACE2, but it's very, very much enriched in the Cory Plexus organoid, which may actually explain why you get far more infection. So using this new protocol, we use the same conditions. And as you can see here, you actually get very robust infection of these organoids. So quantifying at about 24 hours post-infection, you get anywhere between 10 to 15% of the cells infected. And this actually goes up by 72 hours. You get closer to 20%. Then this suggests that maybe the virus is spreading within these cells. So to look at this further, we actually wanted to see if infectious virus is being released from these cells following infection. So we did two things. We sampled the organoids themselves at 0, 24, and 72 hours. We also sampled the culture supernatant. And what we noticed is, at least in the organoid lysate, by 24 hours, you can see a significant increase in the amount of virus, which goes up much higher by 72 hours. But in the medium, you could really only detect this increase in virus at 72 hours. So this suggests that maybe early on, a lot of virus is being produced, and it's being released maybe about a day later. So this really confirms that not only can the virus infect these cells, but the virus can replicate in these cells and be released from these cells. And as I said before, initially we used pretty high titer just to look at whether the cells were susceptible. But interesting in the Cory Plexus, even if we go down to 10 to the 4 and 10 to the 3, you still get pretty decent infection. And so at 10 to the 3 condition, you're getting about 2% of cells infected, and this is still higher than the amount of neurons we saw infected. So this suggests that the Cory Plexus epithelial cells have at least like 100-fold greater susceptibility to the virus than neurons. And interestingly, not only are the cells is the virus spreading by cells releasing virus, but we did see the development of these syncytia. So it's basically when cells fuse together, so you have multiple nuclei in one cell. And this has been reported in the lung as well as a lot of 2D cultures. So really by 72 hours, we can see the formation of a lot of these big syncytial cells. And they've had even as many as 12, I've seen maybe even 20 nuclei in one cell. So this is perhaps another way the virus is using to spread along these cells. And so we know that it can infect the cells and spread. What are the functional consequences? So one thing we did notice using tunnel staining was that the cells that did get infected 24 to 72 hours later did tend to die. So they have these, they're tunnel positive. But the really interesting thing here we noticed was in the Cory Plexus at least, even cells that were not infected near the infected cells were also dying. So this suggested some cell non-autonomous pathway, which we haven't done too much as far as mechanism, but I will hint later at maybe what we think is going on here. Lastly, to get maybe an idea of what's functionally happening in these cells, we did RNA sequencing. And so one thing we did was we could align the RNA sequencing transcripts to the SARS-CoV-2 genome. We saw massive upregulation of transcripts in these cells over time, which is further evidence that the cells are actually producing transcripts. And then we also did a differential gene expression between the mock and SARS treated ones at 72 hours. And as far as what's downregulated here, we can see on the left or in the middle here, you have the geoterms. Then we pull out some specific genes here to show the patterns. Really, the major things that are downregulated are things like ion transport, transmembrane transport, like cell connections, cell junctions. So it's basically indicating that a lot of the normal function of these cells is being perturbed, so they can't generate the same type of ion gradients and things that you would see normally in the CSF. As far as what is upregulated, we saw a lot of increase in these inflammatory cytokines and a lot of extracellular vesicle Golgi processing, which is likely involved in creating more virus. And so specifically for the cytokines, we saw things like IL-32, CCL2. A lot of these have been described in the literature in COVID-19 patients. So perhaps these inflammatory cytokines are creating an inflammatory environment within the brain, which may lead to some of these symptoms. And so lastly, just to get at part of this non-cell autonomous effect that we're seeing. So
we will have to wrap up shortly. So
yeah, last slide. So this we see this the TTR protein that goes down in the infected cells, as well as we notice this one thing, the phospho-P38. So it's part of the P38-MAPK pathway, which has to do with a lot of cell stress that have been reported in other literature. So you see massive increase in phosphorylated P38 in the infected cells, but you also see it go up in the cells near them. So this may help explain why you see increased cell deaths and stress in the cells nearby. So this is just a conclusion slide. I've basically said all these things, but basically what we see is you can infect neurons and astrocytes minimally, but basically the Cory Plexus seems to have this very high neurotropism for the virus. And basically when the virus infects these cells, you have functional deficits that occur related to CSF production and barrier formation. And with that, I just want to thank a lot of the collaborators we've had involved with this. The paper is online now, so you're welcome to scan the QR code. You can read the whole paper. And if you have any questions, you're always welcome to email me. Thank you.
thank you so much, Fadi. Really interesting work, and glad to see that the paper's online. I remember seeing it come out earlier on as a preprint. I just wanted to ask now, circling back, you hinted at this, but what do you now think is the sort of implications of the choroid plexus involvement in the neurotropism for the symptoms that we're seeing in some of these severe patients that you described at the beginning?
Yeah, it's a good question. So we looked into it. So one thing I mentioned, that TTR expression goes down. So TTR is it's a carrier protein, so it's very important in bringing thyroid hormone into the CSF. So I looked into the literature, and decreased levels of CSF thyroid hormone are associated with symptoms like brain fog and reduced cognition. So perhaps this could relate to it, as well as a lot of the inflammatory cytokines that are secreted could help increase inflammation sort of in the brain. But with our study, we haven't really done any in vivo work, so it's hard to necessarily say for sure what's going on. And we don't really answer the question if the virus can actually get there in the first place. We just know if it can be exposed to these cells, it can be infected.
Got it. Okay, very interesting. Thanks so much.